Swayam Prabha. Digital India. Educated India.
Welcome back. In lecture 54, we will talk about artificial sewage treatment. This is part 2, where we will talk about secondary treatment. So, the different concepts that we will cover are on biological or secondary treatment. Then we will talk about attached growth systems, then activated sludge process, then stabilization and oxidation ponds, and finally, on certain points on artificial methods of sewage treatment. So, biological or secondary treatment involves both aerobic and anaerobic microorganisms, and there are different types of biological treatment, and there are several technologies, but principally there are 3 or 4 types. We can broadly categorize biological treatment to 4 types, but of course within this and there are several varieties or modifications of these processes, and that actually has led to several technologies that has been developed over time. So, the first broad type of biological treatment could be referred to as attached growth systems, and in this particular system the sewage is treated with, of course, with bacteria in aerobic condition and using the process of filtration. That means in case of soil, if you remember, when we applied sewage on soil, the pores of the soil acted by prevented the solid material to pass through, and in the pores the sewage was acted upon by bacteria. So, the same principle is more or less utilized to certain extent, of course, but we will explain that. So, in this particular case, we use a filtration method where a large porous beds with some amount of filter media. It could be sand, it could be brick bats, it could be plastic media. So, it depends on what sort of media we use. Based on that, certain other characteristics could be changed. So, this media facilitates actually aerobic bacteria growth, and that means they form this aerobic bacteria grows over this particular the media, and they form a film, and when the sewage is passed over this particular media, this aerobic bacteria acts upon the sewage, and then the sewage gets treated. So, that is how this particular system works. So, in activated sludge process, the next process, here the sewage containing the organic matter is first of all aerated. That means we put in compressed air and inside a particular basin, or we call it a aeration basin, and because we are putting in air, microorganisms grows because in the presence of oxygen, and the metabolizing or the reactions actually increase, and organic matter is actually stabilized by these microbes, and some of the and that actually leads to because we are it's in aeration tank, sludge forms, which could be sometimes like a flock, which is a mix of organic matter as well as bacteria, and all this that is formed, this could be also reused again. So, once that at one end we are actually increasing or reducing the organic content in the sewage, in the other end we are creating this flock or a bacterial or a mass where we have this microbes, bacteria, and then we have some amount of sewage as well, or the suspended organic matter as well, and this actually could be used by recirculating that, putting it back into the influent or the coming in wastewater, so that the growth of bacteria starts from there itself, right. So, that means we are creating a population of this bacteria, which could be used again and again for treatment of this particular wastewater. So, sewage containing organic matter is aerated in an aeration basin in which microorganisms metabolize, and the organic matter and a portion of the sludge formed is recirculated to facilitate further bacterial growth. So, we sometimes recirculation may not be done, we just do aeration, but there is no recirculation, and this could be referred to in case of aerated lagoons. We actually have broad tanks where we let the sewage and then we just put in air through it, but we do not do any sort of recirculation. But broadly activated sludge process refers to systems where some amount of sludge is used, or in the other terms activated sludge is used to start the decomposition process. The third type is called immobilization carriers, and these are basically advanced systems, and primarily this could be referred to as advanced growth systems, where media is in a fluidized state. That means instead of it is a fixed bed, instead the media is in a moving state, that is a fluidized state. So, because this organic because the matter is this media is all over this particular this sewage, because it is moving inside the sewage, there is consistency in organic removal, and it also reduces the sludge volume. That means more amount of decomposition takes place. And there could be different types. There could be like one technique is moving bed biofilm reactors, MBBR, or fluidized aerobic bed, FAB. So, there are different technologies which are involved, which has variations of designs and so on. But roughly we understand that it is a attached growth system, which is like we use filter media, but instead of the filter media being fixed, it moves around in the sewage. So, these reactors or these chambers where this reaction takes place are similar to aeration tanks, and the movement is mostly circular, and air actually helps in their movement, and specific gravity of media allows flotation and movement, because most cases we would be using this HDPE or plastic media. So, they usually float in this wastewater, and that is why they move around in the wastewater. Then finally, we have got stabilization ponds, and these are artificial open flow through earthen basins where the organic matter is in sewage is stabilized. And as you understand, these are natural processes where we just put in the sewage in some tanks, and it takes certain time for the sewage to be stabilized, acted upon by bacteria, and so this takes a certain period. It may range from a few to several days, and sometimes it depends on what kind of pond it is, it may take even months. And also based on the climate, it may take much longer periods. So, let us first start with attached growth system. And in this particular systems, as we have discussed, there is a filtering media, and the treatment takes place at the surface of the filtering media where the film is formed, and usually colloidal and dissolved organic matter is acted upon, and it takes a few minutes in a trickling filter for this matter to get absorbed by the this bacterial film itself, right. So, it does not take much time, and bacteria converts organic matter into stable inorganic forms, and all material is separated or precipitated in biological treatment units, or biological flocks are formed, and these are separated in secondary settling tanks, and this we have discussed earlier. So, let us talk about the media that is used. Usually the media in today's context is a synthetic media, and why this is designed? These are designed media which has got very high specific surface area, then high percentage of void within that media, and this are resistant to abrasion, and they have good structural strength, insolubility in sewage, and resistance to spalling and flaking. So, the media is done in such a way so that it maximizes the surface area where this biological film is formed, and then that means that the more amount of sewage comes in contact with that, and the this conversion or this organic matter stabilization is very, very fast. So, usually it is made of PVC, PE or HDPE. Then looking at hydraulic loading rates of this kind of systems, it comes to around 40 to 200 meter cube per day per meter square, and this is the amount of sewage that could be loaded based on the surface area of that film. And organic loading rates are this is 0.8 to 6 kg of BOD per day per meter cube. So, this is the amount of BOD that could be removed using this kind of system. And how we calculate this? We have already done that in one of our earlier lectures, that how to estimate BOD of the total quantity of sewage and then convert it into grams and see that how much of it could be applied as well. We can also based on this particular rates, we can determine what is the volume of sewage that could be applied in this particular system. So, sometimes we can also have interlocking sheets of plastic arranged in a honeycomb fashion, which also acts as that surface where the film is formed. And this kind of reactor depths or this chamber depths could be around 12 meter. So, sometimes this is synthetic media, but also we can have very simple systems where we can have sand filters or intermittent sand filters, and these are sand beds with open jointed drainage pipes below, that is 90 centimeter to 120 centimeter, 3 to 4 feet below, and this the effluent comes down through the sand and it gets filtered in the sand, and the organic matter is acted upon in the pores of the sand, and then the effluent goes into this under drainage pipes from where it is drained to the next to the disinfection tanks maybe. Then flooding of these beds are done at a depth of 3 to 10 centimeter every 24 hours. So, gradually this is the flooding rate of the tanks, and then automatically it takes 24 hours for this amount of sewage to go down via this particular sand filter. And similar to sand filter, we can also use this other media such as broken stone and brick ballast, and these are known as contact beds, and this filtering media is of 2 to 2.5 centimeter size. Actually, this both sand filters and contact beds were used earlier, and also in some maybe in some parts of our country we still see them, but nowadays synthetic media is utilized. So, trickling filter is one more system where you can see the design of the trickling filter. So, these are the filter media, and this is the effluent pipe which is taken up, and then actually this is a rotating arm, and then this actually sprays the sewage over the filter media. So, this spreads the sewage over the filter media. The purpose of this spreading like this, that means some amount of aeration also happens because of this. That means some amount of oxygen gets mixed with the sewage, and then it goes via the filter media, and then it goes below where it is collected, and from there it is taken via outlet to the next chamber. So, trickling filters are usually preceded by a primary sedimentation tank and followed by a final settling tank, because the effluent that comes out has got lot of scum in it or lot of the slime in it, and that needs to be separated. So, that is why we use a final settling tank, or you can say this is a secondary clarifier. So, as sewage trickles through the filter media, a biological slime forms which absorb the organic material in sewage. So, that is for the initially, and then the biological this matter is already there, and when sewage keeps on moving on it, then one time when the layer is very thick, then it actually peels off and then comes down along with the sewage, and that is why we need to have a secondary clarifier, right. So, that means this scouring takes place when the slime thickens, and then new slime layer actually grows over on that particular surface. So, what sort of loading, both hydraulic loading and organic loading has to be considered. Like the previous case, we have to consider the rate of wastewater application and rate of BOD application, and circulation recirculation ratio. Some amount of sewage could be recirculated, which is 0.5 to 3%. The distribution of this sewage could be done via this particular distribution, this arm, which could rotate at a 2 RPM, rate of 2 revolutions per minute. And we have an under drainage system V shaped half round. So, the under drainage system below is V shaped, and this is covered by larger blocks, so that this smaller filter media doesn't goes into that. Some amount of ventilation could be also put in, that is 0.3 meter cube per meter square per second, and the contact media is 30 mm to 80 mm in size. Filter depth, gradually concentration decreases at lower levels, and for high rate of filter the depth is 1 to 1.8 meter, and for low rate of filter the depth is 2 to 3 meters. So, that means we give lesser treatment in a high rate of filter. So, more water can pass, wastewater could be treated, whereas in low rate of filter we give a higher depth, and the movement of water is also slower in this particular case. Next, we will talk about the activated sludge process. And in this particular process, as you know that we use activated sludge, that means some amount of sludge is recirculated, and that actually activates the this settling process or this decomposition process. So, this is the aerobic process, and so there is a aeration tank, and we use a secondary settling tank after this particular process. And a sludge return line is also there from the secondary settling tank to the aeration tank, and excess sludge waste line is also there through which sludge is removed. So, that means the after aeration, a biological flock is formed, the effluent is the organic material is also acted upon, the BOD is removed, and but this flock that is there, that or this sludge that is formed is taken to is separated via secondary settling tank, and then this goes either this could go to a sludge tank or sludge digestion tank, and some part of the sludge is sent back to the initial aeration tank, so that it can mix with that and provide that initial set of bacteria which would start the decomposition process. So, that is why this activated sludge is actually this process is known as activated sludge process. So, blowing of air in the sewage or aeration results in flock formation which contains bacteria, protozoa, fungi, etc., similar to the slime in sewage filter. So, the principle is more or less same, and this flock settles when air is stopped, and when added to fresh sewage causes its digestion. So, this flock actually acts upon the organic matter, breaks it down, and at the same time this flock, when this aeration is not done, then it automatically settles down, it becomes the sludge, and we can use this some part of this sludge back into the aeration process, so that it starts that biological growth of that in that particular aeration tank and so on. So, raw sewage after primary treatment is to get taken into this activated sludge process aeration tanks, and activated sludge process is actually it's a detention period of around 1 to 1.5 hours. So, that means in the aeration chambers, after primary treatment, where it is the sewage is retained for around 1 to 1.5 hours, then it is taken to a aeration tank, and raw sewage is mixed with activated sludge and sent to the aeration tank. So, that means the some amount of raw sewage is mixed before the aeration process starts, and this is known as mixed liquor, this particular mixed sludge as well as the raw sewage. And in the aeration tanks, the sewage is aerated and agitated for almost 4 to 10 hours. So, it depends on the design of the tank, it depends on what shape of the tank, but roughly it takes around 4 to 10 hours for the sewage to get mixed or mixed with this this organic matter. So, with this microbial matter using oxygen. And finally, after this we understand that this actually results in the digestion or decomposition of that particular organic matter, and removal of the stabilization of the organic matter, and then finally we take this to the final settling tank. And part of the effluent is sent back and mixed with raw sewage before primary sedimentation, or you can say this part of the effluent or sludge actually is taken and mixed with the raw sewage before primary sedimentation, and part of the sludge is returned for seeding to the sewage before aeration. So, that means from after the treated effluent, some part is even could be taken to the sedimentation tank before even before the primary sedimentation process. That actually helps in starting the this biological growth in that particular media, and even though the sedimentation process is not able to remove that biological matter, but we start the biological growth process over there, so that when it comes to the aeration tanks, eventually automatically there is a certain amount of bacterial growth in that particular wastewater. But at the same time, some part of the sludge is returned for seeding to the sewage before aeration. So, that means some sludge is also added before the aeration tank as well. So, both could be done. So, two terms, one is mixed liquor suspended solid. So, this as we say that this mixed liquor is this mixing of sludge with the with this raw sewage is actually known as mixed liquor, and this suspended solid concentration in the aeration tank liquor this is what it refers to, and this index of mass of active microorganisms in the aeration tank, that is given by this mixed liquor suspended solids. So, that gives us an idea about how much is the mass of active microorganisms in the aeration tank. And similarly, mixed liquor volatile suspended solids, the amount of volatile suspended solids give us an idea about aerobic and facultative bacteria present, which are among the microorganisms. So, we get an idea about this kind of bacteria in the this aeration tank. So, these are the two terms which are important and sometimes used for designing, or sometimes used in while in the process of designing this kind of systems. These are the two terms which are used to determine the quantity of bacteria that is required for this stabilization of this organic matter. So, this images shows you certain different forms of activated sludge process. For example, in the first process, you can see it is a conventional activated process. So, we have influent coming in from the primary sedimentation tank, it comes into the aeration tank, then the sludge is formed, which could be, this is a settling tank from where we can take out the effluent from the settling tank. The excess sludge comes to this part, then some part of it goes back and then is mixed with the effluent, some part of it is taken out. So, only a certain part is reused and this return sludge is mixed before the influent goes into the aeration tank, and that actually leads to further removal of organic matter. Then is contact stabilization. In this particular process, there is no requirement for a primary sedimentation as well. So, we can do this particular process without primary sedimentation. Why? So, the influent wastewater is taken into the contact aeration tank, everything else remains same. Only thing is the return sludge is actually first re-aerated, and that means further stabilization of the organic matter happens over here, and that actually reduces the quantity of sludge further, and this is actually used again mixed with the wastewater for further aeration, and this actually helps in the process process of removal of suspended matter as well as this biological matter is much more effective, which we can achieve over here. Because we are already use a secondary settling tank, so it takes care of both the suspended matter as well as organic matter, which also gets deposited in form of sludge. Next, we have oxidation ditch. So, in oxidation ditch, you can see that this is the oxidation tank. So, oxidation tank would be, I will talk about that after this. So, in oxidation tanks, we allow the sewage to be on a earthen basin, and where you can allow it to mix with the air in the atmosphere, and then aerobic bacteria can act upon on the upper surfaces, anaerobic bacteria can act upon at the lower surfaces. But more or less this is where this instead of aeration, mechanical aeration, we can say this is natural aeration is taking place. Everything else is same. That means we from here this excess sludge is taken to this influent is taken to the settling tanks, and where we can get from where the sludge that is generated, some amount is excess sludge which could be taken to landfill site, whereas some amount could be returned back and put it into the influent before it goes into the oxidation tank, and that also increases the digestion or the amount of stabilization of the organic matter. Then we have got step aeration, and in this case you are seeing the influent is coming down through multiple channels. So, basically there are multiple channels in the aeration tank, and usually this return sludge is mixed with those particular at this within the this aeration tank, and because we are taking the influent from multiple channels, there is more thorough mixing, and then not only that the influent wherever it is coming in, here the mixing volume is more, then in the next layer the mixing volume is less. So, usually that is why if we divide the entire flow into multiple channels, then mixing could be more effective. So, this is the same system as we have learned, but only thing the influent is being mixed in a much better way. So, a complete mix plant is where both the influent as well as the return sludge is first mixed and then taken via multiple channels, and then each channel is separate, and then from there we can everything else, then we go to the settling tank where excess sludge is taken out and some amount of sludge is returned. So, this is a complete mix plant, and as you can understand here, mixing is even more than a step aeration plant. So, these are some of the variation of the activated sludge process. Finally, we have got stabilization or oxidation ponds, and as we were discussing earlier, that these are artificial open flow through earthen basins, and they are designed to treat sewage, and usually the detention period is larger and organic matters get stabilized by natural forces, not by mechanical aeration, but by natural aeration, you can say. So, these ponds could be both aerobic, anaerobic or facultative depending on the mechanism of water purification. So, aerobic, we understand that oxygen is actually used. It is used for algal photosynthesis, that means photosynthesis of the algae, and usually this kind of ponds are shallow, that is only 0.5 meter deep, and usually start to prevent anaerobic conditions in settled sludge. So, we do not allow the sludge to settle down. And BOD loadings are around 40 to 120 kg per hectare per day. So, this kind of BOD loading could be taken care of in this kind of aerobic ponds. In anaerobic pond, purification is due to methane formation, that means anaerobic bacteria acts upon the sewage, and then that is why we form methane and also CO2 of course. So, the depth is larger in this pond, 2.5 to 4 meters, and usually they could be used for treating industrial sewage and in sometimes used for digestion of STP sludge as well, and depending on the temperature and waste characteristics, BOD load could be 400 to 3000 kg per hectare per day, which is much higher than the BOD loading of aerobic ponds, and 5 to 50 day detention period results in 50 to 85% BOD reduction. So, the detection period is even in months, instead of days. So, the problem with anaerobic pond is there is bad odor, the methane formation takes place in the lower levels, and so this sometimes it's whereas aerobic ponds does not have any kind of bad odor and all these other issues. So, it is better to have a system where we can use both these processes, so that we can achieve the 85% BOD removal of anaerobic systems. That means, if we combine both the systems, the effectiveness of BOD removal will probably increase. So, that is where a facultative pond comes in, and it acts aerobically at the surface, while anaerobic conditions prevails at the bottom. And aerobic layers actually prevent the odors, because the upper layers are aerobic, and this CH4CO2 is H2S and CO2 is formed. These are taken by the the plants or the in algae. They take the CO2 and then release oxygen, which further helps in photosynthesis, which further helps in this this aerobic stabilization. So, facultative ponds are the best system that is possible, and here the sewage enters the pond, suspended organic matter in the influent and bioflocculated colloidal organic matter settles to the bottom. So, that means in any kind of stabilization ponds, there is sedimentation, and there is both chemical sedimentation, naturally occurring chemical sedimentation, that is bioflocculated colloidal organic matter, actually those also settles, and in absence of dissolved oxygen at the bottom, that is why we see that anaerobic reactions takes place and CH4 is released, which actually also indicates that BOT is also removed. So, in the upper layers, algae grows, which utilize the CO2, as we were talking about that, and for photosynthesis, and that releases O2, which results in aerobic conditions, and in the upper layers this oxidation happens due to aerobic bacteria, and this interrelationship is known as algae bacteria symbiosis. So, that is what is illustrated in this particular image. So, the other thing that is important is, that means as we have learnt earlier, that even when we are talking about this cell purification process, that temperature, sunlight, this plays a big role in this kind of treatment processes. So, in India, because we are at a higher latitude, we are at latitude from 8.4 degree north to 37.6 degree north, the permissible organic loadings are much higher than colder countries, or at other latitudes which are at much higher latitude. So, in this you can see that the more this latitude degree is less, that means more we are towards the equator, the organic loading is high, because the sunlight availability is also high. So, here the organic loading is 325 kg of BOD per hectare per day, whereas for at the 36 degree north latitude, the BOD loading is around 150. Maximum BOD loading that we can give is 150 kg per BOD of per hectare per day. So, that is that means the amount of sunlight or the temperature also plays a big role in determining what kind of BOD loading we can give for this particular stabilization ponds. So, oxygen produced by photosynthesis also depend on sunlight. That is why sunlight is important, and temperature is important. As I was discussing, that in India BOD removal is almost 4 times compared to colder countries, and the depth of these facultative ponds are adapted around 1 to 1.5 meters, which is in between aerobic and anaerobic ponds, and detention period is about 6 days, and around 80 to 90% of BOD is removed by treatment via this kind of ponds. So, as you understand that the detention period of 6 days means we have to design this pond so that they are able to take the incoming sewage for 6 days. And not only that, this ponds needs to be dislodged at intervals of 6 to 12 years. That means there is lot of sludge that gradually settle, and this that and accumulate, and the rate of accumulation is around 0.07 meter cube per capita per day. So, you also need to multiply the number of people for whom this wastewater is generated into the number of years, or like if the discharging frequency is 6 to 12 years, that means you have to multiply by 6 or 12, and with 0.07 meter cube, which is the amount of sludge that is formed, and you will get the volume that is required to retain the sludge. So, if I have a tank, the lower part is for sludge, storing this excess sludge, which is 0.07 into population into number of years of cleaning. So, that will actually give you the sludge volume, and the upper volume is for detention period of 6 days. That is total wastewater, incoming wastewater, that is Q multiplied by 6 days. Okay. So, this is so that is how we can determine the volume that is required for this kind of stabilization ponds. So, some protection measures has to be taken. It has to be around 200 meters from residential areas, because wind will carry the smell from this pond, and also better to have drinking water wells at least 15 meters away from the pond, so that there is no contamination and all. And usually these ponds are rectangular with length not exceeding 3 times the width, and maximum basin length is sometimes kept at around 750 meters. So, you can see in the images the designs, there could be several arrangements. Over here these are arranged in sort of series, one after another. Here it is also we are divided one large pond, and then there are 2 small ponds. Sometimes it is applied all parallelly to all these ponds together. So, there could be different arrangements of this particular units. So, ponds are designed in multiple units in either series, with the first pond having 65 to 70% of the total area to avoid anaerobic conditions. So, the first pond is actually bigger, as you can see in this particular image. It holds 70% of the entire volume, and that is done so that we can have a larger pond, and if it is a smaller pond, there is more chance of this this anaerobic conditions developing. So, it is better to have the first pond larger, because most of the sewage load comes into that, and then from there we can have subsequent ponds which are smaller in size. So, we can have multiple cells in this system, as you can see 1, 2, 3 cells, here also 1, 2, 3 cells, and this could be in parallel, and if it is parallel, then it facilitates maintenance like dislodging or repairs without upsetting the entire process. So, that means we can keep 2 operating while 1 is being repaired, and parallel systems also provide better distribution of settled solids and multiples, but also we have got we can have multiple cells in series, which decreases dispersion number and enables better BOD and coliform removal and reduced algal concentration in the effluent. So, series are more effective in BOD removal and reduced algal concentration in the final effluent. So, in some ways series are better, whereas for if we do a parallel treatment, then in terms of maintenance and all these things it becomes much easier. So, individual cells also should not exceed around 20 hectares in area for this kind of stabilization ponds. So, these are the different methods of biological treatment, and now overall if I look into the entire sewage treatment process, there could be several ways we can arrange the different treatment units to effectively treat the sewage for a particular area. So, we have primary treatment, we have secondary or biological treatment, then we have got final treatment. So, in this particular image you can see different two options. One is the first one is you can see this part is the primary treatment and this part is the secondary treatment, and aerobic, this is where we are using aerobic mechanized biochemical sewage treatment process, and secondary treatment can also be extended aeration and without digester. So, we can do more aeration, that will reduce the need for a digestion, further digestion tank at the end for sludge of course. So, that means we can use variations of the same system and we can and that actually has to be determined based on lot of factors, based on the type of sewage and based on the overall arrangement of the treatment unit and so on. Let us see this first one. So, we have raw sewage, then we have raw sewage, then we have a screen, and then the grit chamber where grit removal takes place, and then we have the primary settling tank, the first one. Then we have our biological reactor, which is the biological oxidation and synthesis happens in this particular reactor. So, this could be an aerobic mechanical system, and then we have our secondary settling tank, and then we get our treated sewage which can be taken out and disposed of or reused even. And we have a secondary sludge tank which is brought to a thickener where the sludge is thickened, that means the water is taken out of the sludge, and then some amount of sludge could be taken to the primary settling tank. This supernatant, that means the water could be taken to the primary settling tank, whereas some sludge comes from the primary tank to this particular thickener, and this thickened sludge actually is taken to the anaerobic digester, and where the sludge is being digested, and again supernatant, which is basically the clear liquid that can be taken and put into this particular before the primary settling tank, whereas the digested sludge could be taken out for final disposal. And this anaerobic digester, we have CH2 and CO2 gas coming out of that. So, here we are not having a activated sludge process. So, this is just a biological reactor, maybe it is not a aerobic mechanized system. This could be a particular this intermittent sand system or a contact bed or any other kind of media could have been used. So, this is a attached growth system, and this is the entire process, but we could, instead of this, we could also have a aerobic mechanical biochemical sewage treatment process as well, and then actually we can have to have a rich sludge return line which will probably go into that particular aeration tank. So, this is another system where this is a conventional aerobic sewage treatment for including sludge digestion is not there, which it has to be added. So, we have this screen, grit chamber, and then without any primary treatment we can directly have this kind of treatment where anaerobic reduction and synthesis can take place, where we can have an anaerobic filter or we can have anaerobic RBC. This is then we have a UASBR, and so that means this is upflow, then sequential batch reactor and so on. So, there are several processes that could be involved where we can get rid of the settling tank, and depending on how much amount of this digestion takes place, and then we have this final settling tank where thus both the suspended solids as well as that excess deposited sludge from the organic matter as well, all could be separated. So, depends on how we actually organize this particular system, depends on what kind of treatment we actually devise. So, this is where we can say that only biological treatment is also adequate. So, as to we can just do a primary treatment and let the sewage out. Similarly, we can only do biological treatment and let the sewage out as well. So, these are two more systems, two more arrangements of sewage treatment plants, where you can see that we are using reusing the water for some amount of irrigation or food crop irrigation. So, we have untreated wastewater, we have screens, then we have grit removal, some amount of aeration is also done over here, and then we have the primary sedimentation tanks, solids goes to the digester, this is the sludge digestion tank. Then we have a trickling filter, then we have again having a aeration and a flocculation basin, we have then a clarification, secondary clarifier is there, and then finally we can get rid of some of the this effluent over here, and then we can also this after clarification we can have this flocculation, filtration, then we can have chlorine as well. So, that means we can have some amount of once the clarification is there, then we can use chemicals for flocculation to some extent, then filtration is there, we can add chlorine, then we can have then we can use this for storage, and we can store it and use pumps to do for this food crop irrigation. So, this is again you can see that we are using chemical coagulation after even a biological process. So, these are specific case specific arrangements that are done in particular cases, because we require maybe some amount of chemicals are still there which has to be removed only via this particular process. So, in the next one you can see that untreated wastewater, screens, primary sedimentation, oxidation ditch is used instead of trickling filter or a biological treatment. Instead of a artificial biological treatment, we use a natural biological treatment. Then again clarification, secondary clarifiers, then some amount of water is discharged, but the water that is reused, that is again taken through a ozonation tank. We put in ozone over there, so that is sort of disinfection happens over here. So, we have again clarification over here, some chemicals are mixed over here, then sand filtration, then activated carbon absorption, UV disinfection, and finally again agricultural irrigation. So, this there could be more complicated ways of treating wastewater, depends on how you are going to finally use it. Based on that we would do the treatment. So, to conclude, sewage can be treated via various combination of processes. This is decided based on quality of discharge intended, space availability and cost, and secondary treatment can be directly applied after removal of grit as well, and oxidation ponds are easy to implement provided adequate space is available. So, these are the references that you can use. Thank you.